I'm going to talk about our applications using digital PCR in terms of methylation-specific PCR. And I'm going to tell you a little bit of the background of the science and a little bit of history as to why we're excited about the technology and why we're doing methylation analysis on blood cells. And essentially, I'm a molecular epidemiologist and have been working on understanding causes of cancer and why people get cancer and how they respond to therapy for many, many years. And one of the things we've been always interested, especially in neurological surgery in terms of brain tumors, is the influence of the immune system. But in my lab, we have 12 or so -80 freezers, and over the last 25 years, we've collected thousands and thousands of samples of blood from patients with various cancers, but certainly a lot of brain tumor patients, and also carefully selected controls from around the Bay Area. And so one of the motivations for this whole topic is that we really would like to be able to probe the immune system using archive samples. These samples were never collected to be used for immune analysis. There's no cells left. They're just whole blood frozen in your freezer. So what's there? It's DNA from 20 years ago. And we have lots of samples, and so we'd be very interested in a technology and an approach that we could use that would allow us to access those archival samples. And a few years ago, and I think I've got something a little bit more of a historical presentation here. A few years ago, we were aware of studies coming from Germany on an approach which seemed, frankly, sort of radical, and that was that you could, they claimed, this group claimed that you could look at the promoter of a transcriptional factor called FOXP3, and the methylation status of that promoter was very specific for a very important kind of immune regulatory cell called T regulatory cells. And that kind of got us thinking about methylation as a very specific mark of a lineage within a lineage of cells. And then a few years later, postdoc, now assistant professor at Dartmouth, Brock Christensen, did this study and published this in PLOS Genetics. And we were just starting to use the array approach, the Illumina arrays, for doing methylation analysis in tissues. And we just got the idea, let's just look at a bunch of normal tissues and see what they look like. And, you know, not a sophisticated hypothesis-driven experiment, but it turned out to be quite interesting because these are just normal tissues taken from our freezers and some of our collections of stuff that we have on hand, and we were able to actually completely distinguish all these tissues with fairly high degree of statistical confidence simply by looking at the methylation patterns. And that gave us this idea that maybe methylation could be a signature, and also led to what I now call a barcoding concept for lineage differentiation by DNA methylation. And I now believe that this is sort of a proven principle, that cells, especially we've looked extensively in the immune system, and I'll show you, cells have a unique DNA methylation pattern, and some of those differentially methylated regions are highly specific for a particular kind of cell, like T regulatory cells. But we've now found them for natural killer cells, CD8 cells, CD4 cells, you name it. There's going to be some set of genes that are going to be differentially methylated that could actually be used if you could quantify them very carefully to count the cells, to enumerate the cells. And so now you can kind of see where I'm going. We've got those archive samples with all that DNA that we've been saving for so many years. We may be able to go count using this principle, the methylation, the numbers of methylated and unmethylated target genes that describe different immune cells, and we might be able to profile the immune system just using DNA. No proteins, no cell surfaces, no intact cells, just DNA. And that's really what led us to digital PCR, although I'm just going to show you a lot of this stuff is array-based, and what we've kind of used the digital technology in qPCR in a confirmatory way for these arrays, much as what you've heard earlier in other talks. But they're also interesting standalones. But I didn't mean to turn it off. But and I'm just going to give, in case you are interested in this, there is a mathematical problem. There was a sort of a difficult problem in terms of the arrays that had to be overcome, and that is that if you have these patterns all mixed together of cells, how do you disentangle them to actually get counts and come up with profiles? And that was it's a mathematical problem, and it was solved by our mathematician who's now up the road at Portland, Andy Hausman, and this is published in the BMC Bioinformatics a couple years ago. We don't have digital arrays yet. We're waiting for you guys to make them, because we're really going to be interested in those, because they'll be, I think, super important. So let me then try to work into, and so here's some of our work on the immune system. Again, this is array-based data. The unmethylated promoters are shown in yellow. If it's methylated, it's blue. And what we did is we took whole blood from normal individuals and we sorted individual cells, and you can see what this type of cells that we've separated out in this particular experiment. And then we individually run them on the arrays, and then we find these regions which denote what we call differentially methylated regions. And so in here in the T cells, you can see that that's one. And then we've also found some differentially methylated DMRs for NK cells and B cells. And you can do this with all sorts of cells. And we've done, I'll show you our data with some of the T cell data for the digital analysis. But we've already applied digital to NK cells as well. And here's just a paper that we did using quantitative PCR, and this was sort of, and what we're showing you is just, here's a gene that we found in that previous hierarchical clustering that you saw that describes T cells, all T cells, and a few NK cells. It's called CD3Z. It's a component of the T cell receptor. And the CpG island is highly demethylated in T cells and completely methylated in everything else that we've looked at. And we did, our standard curve using quantitative PCR, and then we took the blood and ran it through a FACS analysis and did a T cell count, and we do get a nice correlation between the counts measured by qPCR and the FACS analysis. And this is tumor. I'm not going to talk about the tumor studies. But in the process of doing this paper, we got really frustrated with qPCR. And the fact that over time, when we do a lot of experiments, these are different individuals, and we're trying to keep the standard curve to behave itself over time. And at low copy numbers, we got a lot of, we have a lot of noise, let's just say. And so that's why, this was a couple years ago, and so we were really happy to be able to try another approach, and that's what I'll show you. And I won't show you the actual data here. This is about brain tumors and T cells, which is an interesting story by itself, because patients with high-grade gliomas look like HIV/AIDS patients. Actually, they have very few T cells, and it's a very highly immunocompromised disease. But anyway, we showed that with qPCR and can show it with FACS and flow analysis. So what is unique about this approach, and what you have to understand, is that all these techniques, or the major techniques for methylation-specific PCR, are based on this bisulfite conversion. And I don't know if you're familiar with it, but methylation at CpG sites doesn't alter the base pairing properties of DNA, so you have to induce some sort of a chemical difference that is differential with respect to, that will be recognized, and that is achieved with this bisulfite conversion process. Anyway, long and short of it is a somewhat destructive process to the DNA. You're removing this amino group, and then you add sodium hydroxide, and you cook it overnight, and you wind up getting uracil, and then in a PCR reaction, this will base pair not with guanine, but with adenine. Now you have a way to design primers and PCR methods which are specific for methylated PCR, methylated DNA, because if there is a methyl group, it blocks this bisulfite transition. So methylated DNA is resistant to this sodium metabisulfite conversion. You don't pull off those amino groups. The methyl group blocks that reaction. So it's a differential chemical reaction. Anyway, the important thing, I think, for us in terms of PCR is that that degrades the DNA. You're cooking the DNA overnight at 65 degrees with this quite a toxic chemical, sodium metabisulfite, and then you're adding sodium hydroxide, and so the DNA comes out fragmented. And the PCRs that are based on this, you no longer have base pairing. You no longer have duplex DNA, because what you've done is you've taken all the C's that are unmethylated and converted them to U. So they don't base pair anymore. So the PCR is all single-stranded PCR. And that is another reason you get into trouble with using quantitative PCR, because you're lagging, only one strand is being amplified. So you're lagging a CT right off the bat when you try to do a standard curve. So enough of that. I'll just show you some data, and then I'll get to, I think, an important conclusion about this. With the help of Dave and the other folks at Bio-Rad, we have tried different things to optimize this bisulfite PCR and using the digital platform. I'm going to just cut to the chase. A lot of this stuff doesn't really matter. The one thing that's, for instance, we talked about enzyme digestion. I don't think that really matters much. What seems to matter a lot are annealing temperatures, not surprising. I'm not going to talk about the monoplex and multiplexing, but we have issues with that. It's not a given that things are going to work together when you put two things together. And again, I don't know how much of this is related to the fact that it's bisulfite DNA, methylation-specific PCR, or I just don't know. But here's just an optimization experiment, and you can see raising or lowering the temperature, you get, and this is a T-cell marker, and so this is a PCR that will only amplify the demethylated promoter of this gene, which is integral component of the T-cell receptor, and that only occurs in T cells. So these are T cells, and they're showing themselves, and you can see we've done our, we don't use the auto thresholding very much, because we got this, we call it rain. I don't know if it's rain, but you can see that the separation becomes much less clear as you reduce the temperature of the reaction. And so, well, you could just threshold this along here. One of the things we've done, though, is we've measured the actual this is whole blood, T-cell concentrations in the blood. And so we're able then to look here. This is actually measured. It's 25% T cells based on flow cytometry and FACS analysis. And so we're able to kind of see that, well, at these lower temperatures, if you include, if you would include this number creeps way up. So we base sort of an external validation. We can kind of recognize that we should run this reaction at a higher temperatures, something like 60 degrees rather than 56 or lower. And here's another gene, the FOXP3, which you can see. Somehow this got on the same slide. I didn't mean it to be on the same slide, but here's another promoter that detects a subset of T cells called T regulatory cells. And here you can see that changing the temperature doesn't really influence this kind of, I don't know what it is. I'd be glad if you guys could tell me what this is. We can threshold this and get very nice numbers, but here we don't have that problem. So actually, the height of this signal for our T cells actually goes up as we lower the temperature. So we feel like this reaction would be best run up in here at a lower temperature compared to the T cell. And anyway, this gets back to the multiplexing. The probes and PCR reactions don't always optimize to the same annealing temperatures, and so it's not always possible to do that. If you just look at isolated cells, here's just a curve for T cells, and here's our curve for the T regulatory cells. These are quite interesting cells. They only constitute about 1% of your blood cells. And so we're getting down here to being able to see one copy per microliter, I think this is. Yeah, 1 copy per microliter. And that's pretty good. That means we can detect the levels, the dynamic range in blood is between 0.1 and 2%. So we're down in that low range, and the digital PCR has the required sensitivity. So we did a lot of studies with the qPCR, as I indicated before, and so we came along and we decided, let's just see how the digital compares to all the data that we generated before. And you can see that this is just showing that, yeah, it's pretty good. The qPCR is a little bit higher, but and the correlation, it's definitely significant, but certainly the two assays are giving us similar kinds of data. And here's a comparison of this, and I want to just point out that you can see right here that T cells are about 20% of your blood cells, but go to these T regs, and you're down here at a much lower level. So we're 10 times lower level, and you can see the noise is increasing, and the difference between the qPCR and the digital gets bigger. The regression gets much noisier and sloppier. So it's a reflection of the fact that we're now looking at, instead of 20%, we're looking at 2% or 0.2% and trying to detect that. So here's kind of one of my take-homes, which I'll leave you with, and which I think is, for us, is important. And that is, we've just shown that digital and qPCR are quite comparable with respect to methylation-specific PCR. But with respect to what we're trying to do, and that is do immunology studies, which is better? Is one better, or are they just equivalent? And the answer is we think that the digital is better, and that's based on this data. And so for all those folks here, and you see these are a lot of different people that we've drawn blood on. I think we have, I don't know what's in this. We've done a couple hundred people. This may be some subset. And we did a flow cytometry for T cells, and we've just regressed the flow number, which is our gold standard. That's what we accept as the truth. And then we have plotted over here the relationship with either digital, which is in blue, or within qPCR, which is red. And there are a couple of things. The regression line has a slope that's nearer to 1, right, for digital compared to qPCR. And the noise around here, the tightness of that regression, is much better for digital than it is for qPCR. So this is gold standard validation of this immune biomarker. And I don't think there's no other data like this that I'm aware of. And so we're of the opinion that this is a superior method. And it's probably, we think, because it's more stable. We did measurements over time on a single individual, a poor person that was, well, actually, this is only 11 samples. He was studied a lot more. But and here we're just kind of comparing digital and qPCR. And I'll kind of get to the end. Here's FOXP3, again, much lower levels. of these cells. Here's the digital. You kind of see how much more stable it is. And then here's the qPCR. And again, every one of those points represents a different experiment, a different standard curve, and which we didn't have to run on this. And I'll just show you this table, which again kind of gets to this point of digital being more stable and a more reproducible system. And here's the three measurements, quantitative, D, and flow. And this is a different set of data. This is a person measured over time. They are different samples. So his blood cells could be changing over time. But nonetheless, the flow measurement, the coefficient for T cells of variation for T cells was about 14%. And you can see digital 17, Q21. When you go down to this, say to this very uncommon cell, but very important, the T regulatory cell, you get a lot more variability with the flow, and similar to the ddPCR, and a lot more with qPCR. So bottom line is that's kind of where we're at. This is T cell data. We have natural killer cell data. And we think this is a general model that we can use either to confirm our array-based data where we see differences based on arrays, or just as a standalone when we're looking especially at these rarer subtypes where the frequency is low and we've got archival blood, and now we can go into our archive and go back 20 years and ask, well, do those patients that we studied 20 years ago, did they have fewer T regulatory cells compared to normal controls? And we have that. We have all that stuff in the freezer. And we also have other diseases, lymphomas, breast cancers, lung cancer, etc. And so it opens up this whole area, I think. And so we're quite happy with it. And here's my colleagues. And this is also a transcontinental analysis. My friends and colleagues at Brown, Dartmouth, and also at Minnesota. And here's our group up at UCSF. And that's not a very good picture, but that's Hawaii from a satellite. So thank you. Yeah.
So epigenetics-based assay design is difficult because anytime you have a CPG, if you don't have 100% methylation, you can get, a site where you have, a couple CPGs and there 50% at one locus is methylated, not the same 50% is methylated
Right.
at another position in the exact same amplicon area. So given that, and given that Bio-Rad's working on the next generation, which is the QX200, which allows you to do just primer-based locus detection, where you can now have a smaller footprint and only have 2 primers rather than the added specificity of the probe. Is that something that would interest you as a researcher, or do you still want to stick with the probe-based approach for your targets?
I think I don't know actually offhand. I know it's kind of like cyber green, right? That's what the gentleman was telling me. I don't know. It may have to
footprint by good
right
significant amount. So because it's so fragmented, that's an advantage, and but you do have a little bit less specificity, so there's a trade-off there. Right,
and we should try it. I don't offhand know if that's going to be the critical part. It's really where if you can get good primers, whether there's a probe or not, it might not matter, and the probe may actually be a good thing. But that's a really, really good point. And all of our assays are based on demethylation. And what I know this is getting a little bit in the weeds, but the heterogeneous methylation that you're talking about is very, very common, and that really makes quantitation difficult because you can't because it is heterogeneous. But in terms of these differentiation markers within lineages of cells, we find that this demethylation is really part of the developmental program, and it's very uniform. So it's just a little inside the. We learned this over a long period of time. All of these are demethylation assays. They are specifically demethylated in the target cell. We don't try to measure the methylated signal because the methylation is, I think the cells don't spend a lot of energy maintaining methylation in a very, very concentrated sense. The real important thing is getting things demethylated and keeping them demethylated. If that gene is the T cell receptor, which is central to the cell's function in life, you want to make sure that that receptor is completely clean, and so you get in a very nice template that's demethylated. Anyway, that's a really good point, and it's worth trying. I for sure. I think.
I wonder if I could add a little bit, because I work on T-Rex biology, and if you really want to know the epigenetic status of the locus, then you sequence, because there are 14 modified sites, and it can be, like you said, muddled and it's mixed. But if you really are interested in the identity of the cells, then I would like to have as many coverage as possible. Instead of the 3 CPG sites, I would like to see maybe more than half of the 14 to be sure that's actually a true Treg.
Yeah. And then there are different regions. I mean, it's
Yes.
picking the DMRs is, because you have to use biology. You just can't. Being able to obtain a pure population of Tregs is a hard thing to do, just by itself. That's what you do. Well, we'll talk to you later.
Yep.
Anyway, thanks.
Thanks again for coming, John.